Hello and good afternoon. I'm Sean and I'm delighted to have this opportunity to introduce you to our company and the services we can offer to you. Medicilon is an integrated contract research organisation providing comprehensive one-stop services from idea to IND application and API manufacture. We only provide CRO services to clients. We don't run our own internal drug discovery projects to avoid any potential conflicts of interest. These are the topics I'm covering for you today. I'll begin by introducing the company, then I'll focus on our discovery chemistry and biology contract services. Next, I'll review briefly our platform technology services for PROTAC, antibody drug conjugate, and nucleic acid drug development. I'll introduce our candidate development, preclinical development and manufacturing services. And finally, I will explain why I believe working with Medicilon provides an excellent opportunity and option to develop your pipeline of new therapeutics. Medicilon is a leading Chinese CRO, delivering R&D support to our customers for over 19 years. Our clients include major pharmaceutical companies such as Roche, GSK and AstraZeneca, SMEs including biotech startups, research institutes and universities. With over 19 years' experience in drug development, we have built comprehensive integrated platforms covering small and large molecule drug discovery. Alongside candidate development and selection, Medicilon can supply a range of candidate optimization, GLP toxicology, as well as formulation and process development activities. We have established a local office in Cambridge to support you by ensuring rapid and effective communication. Medicilon can deliver a one-stop drug development service from idea to clinic. We run a full set of candidate discovery and drug development services. Our GLP and Vivo laboratory is audited and approved by the FDA, and so far, over 260 IND projects supported by Medicilon have successfully entered human clinical trials. Throughout its 19-year history, Medicilon has respected customer intellectual property. Protecting your IP is an integral part of how we work, and in 19 years we have had no significant issues. IP protection is overseen by Medicilon's Intellectual Property Oversight Committee, consisting of senior company colleagues. We ensure that our computer systems are designed with security in mind. Our colleagues all have confidentiality agreements in place, and access to confidential information is restricted. IP protection compliance is monitored regularly. Lab data, information distribution and compound shipping are all carefully controlled. Colleague training is central to our process and is supported, reviewed and developed continuously. But as well as protecting your existing IP, we also work with you to support the development of new IP for your company. Our chemistry drug discovery department is one of Medicilon's major strengths and works closely with our GMP manufacturing expert teams. We have a large and highly experienced team of over 1,200 chemists with over 13,000 square metres of laboratory space, and also with active plans to grow this further. We offer customers a broad range of chemistry services and integrate these with our biology and drug development departments to ensure the candidates we develop for you have the best chance of success. In our pharmaceutical chemistry department, we design novel chemical entities via diverse approaches, including by intelligent design and by more traditional screening campaigns. We facilitate structure-activity relationship determination through rapid cycles of design, production and testing. We're very experienced in a custom synthesis of intermediates, metabolites and radiolabeled compounds. Medicilon has the capability and capacity to deliver scale-up from milligram quantities to over 200 kilograms of pharmaceutical-grade active pharmaceutical ingredients, and we can deliver all IND-enabling documentation for your regulatory needs. Medicilon has developed expertise across multiple platforms and continues to build capabilities as we move forward. Here are just some of the areas of our expertise listed in this slide. They include heterocyclic and macrocyclic chemistry, peptide chemistry, and antibody drug conjugation approaches. We have significant expertise in continual flow and green chemistry production process techniques. We also have expertise in carbohydrate and biocatalyst chemistry. In short, we can meet your synthesis process development needs from early discovery through to GMP pharmaceutical production. We have a strong team of over 800 biologists working across assay technology platforms and in vivo model types. All of our biology colleagues are aligned to chemistry and protein production departments and closely located to minimise cycle evaluation times and to shorten your projects, thereby reducing cost. We're well placed to support you, either for small focus projects such as testing compounds in an established in vivo oncology model, or for delivering innovative, fully integrated projects for your company. Our biology teams can express, purify and evaluate your proteins of interest via a number of expression systems, including mammalian, E. coli, yeast and baculovirus. We use CRISPR-Cas9 gene knockout and in technology, as well as traditional approaches to establish new cell lines for drug discovery purposes and assay development. We run a range of structural biology approaches to support intelligent drug design and development, and also to characterize your candidates fully. We develop and run a broad range of biochemical and cell-based assays to establish efficacy and structure-activity relationships, as well as evaluating off-target activity for your compounds. Medicilon integrates biology and chemistry in our molecular modelling team to design novel structures, undertake virtual screening, and to design optimized lead compounds. We use continual cell, primary cell, and organoid models and have significant immuno-oncology capabilities. We bring a range of DMPK services into drug discovery early to de-risk your candidate selection. For each project, Medicilon delivers a fully characterized main candidate and a backup from a distinct chemical series. We assess a range of metabolic stability, drug interaction transport and permeability parameters, and this early in vitro to facilitate selection of the best large molecule or small molecule candidates to take forward. We can also undertake method development and validation for bioanalysis approaches. We have over 600 DMPK and toxicology scientists. We also have over 100 scientists assessing efficacy and over 20,000 square meters of dedicated lab space for this purpose. Medicilon can supply a wide range of in vivo models for customers, delivering all aspects of establishing efficacy, safety, and DMPK characteristics. We can bring in vivo assays early into our integrated projects to support selection of druggable candidates. For toxicology studies, we offer both full GLP services and non-GLP rapid toxicity evaluations to minimise costs and to reduce project risk moving forward. Medicilon can supply a wide range of in vivo efficacy models for our customers. We do have a significant focus on oncology, including xenograft and syngeneic models, and you'll hear much more about these models later today. Our in vivo models are linked to our in vitro biology department, where we can undertake a broad array of support assays. We also run a broad range of other disease indication models, including CNS, inflammation, and cardiovascular relevant indications, amongst others. Medicilon has the capacity and capability to develop models for you, and we continually add to the range of in vivo assays that we are offering our customers. We have established significant expertise in designing and delivering more complex studies, including testing combination therapy approaches for synergy. We run a range of in vivo DMPK services, with only some shown here. These assays are fully integrated into our drug discovery programs. Beginning with in vitro assays, we then employ more relevant in vivo models in a range of species, and we work to ensure that the candidate series we deliver for you are tractable. We can use a broad set of delivery routes and sample collection approaches. Our services investigate drug plasma dynamics, excretion, tissue distribution, drug-drug interaction, and analysis of drug metabolites. Medicilon can introduce in vivo DMPK studies early into the process if our colleagues and our clients wish that, so that data can facilitate lead series and candidate selection. We can undertake rapid screening for candidate selection or full GLP IND enabling studies for candidate and backup characterization, moving to the clinic. Medicilon can deliver a complete IND enabling GLP toxicology package to facilitate your clinical trials and regulatory applications. Medicilon's team of experts can help you to plan an appropriate GLP toxicology strategy to support a successful IND application for minimum time and cost. We run a broad range of single and repeat dose toxicity studies. We run genotoxicity assays and reproductive toxicology evaluations. Medicilon's expert teams can also deliver complete histopathology and clinical pathology and evaluations for you, and these areas are particular strengths for our company. Our in vivo efficacy, DMPK and toxicology services can be applied across all candidate modes, from small molecule candidates to antibodies and peptide therapeutics. Essentially, our drug development assays are agnostic to the type of therapeutic you are developing, and we can service a very wide range of projects. We use a broad range of species, shown here, and have significant dedicated laboratory rooms and animal capabilities. Because of our significant capacity and capability, Medicilon can run your GLP studies rapidly to maintain our efficacy and efficiency through the discovery process and to minimise your project downtime. Now I'll introduce 3 of the candidate modes we have significant experience working on at Medicilon: PROTACs, antibody-drug conjugates, and nucleic acid drugs. These, alongside our small molecule development, are representative of the diverse types of projects we can support, but this is by no means the extent of what we're capable of delivering on for you. Although highly successful, standalone small molecules can only target between 13 and 15% of available protein targets. PROTACs, however, have increased the number of potentially druggable targets by driving the degradation of the targeted protein and therefore working as a functional antagonist of its activity. For example, along with allosteric modulators, PROTACs are enabling phosphatases to be directly and effectively targeted for the very first time. This slide describes a basic mechanism of action for a PROTAC. The PROTAC itself is built from 3 units: a specific ligand binding region that targets the cell protein of interest, or POI, an E3 ligase ligand that binds to the E3 ligase, leading to degradation of the protein of interest in the proteasome, and a crosslinker to join POI and E3 ligands optimally together. Construction of a PROTAC from these different units can generate significant potential for molecular diversity. Medicilon has built significant expertise in the chemistry needed to develop effective PROTACs, in the in vitro characterization of PROTACs, and in the in vivo models required to assess efficacy and ADME for candidate selection. We have over 8 years of experience in developing PROTACs. We have over 30 chemists working on 30 active projects at present. And utilising over 300 linkers and multiple E3 ligands, Medicilon has developed PROTACs to target many proteins, only some of which are shown here on the bottom right-hand side of the slide. We begin projects by designing and synthesizing prototype PROTACs and taking these into our in vitro assays to confirm target degradation and also to establish that degradation is mediated by the E3 ligase. We establish cell lines using GFP reporter technology for screening, and we generate a specified number of new PROTACs and assays for a range of pre-agreed parameters with our customers, indicative of activity, of course, and including binding, affinity, E3 ubiquitin ligase activity, and target degradation assays. We also assess a range of properties to facilitate candidate development later on. For each PROTAC, we undertake a series of biochemical assays to assess formation of binary and ternary complex for the target protein, PROTAC and E3 ligase. We use time-resolved fluorescence energy transfer assays and alpha screen technology to assess ubiquitination and degradation potential. Surface plasma and resonance kinetic analysis is run via Biochemical technology to confirm complex formation and to facilitate PROTAC optimization. Our team will confirm target E3 ligase binding via nanobret and nanobit assays and demonstrate target degradation using high-bit, Western blot, ELISA and/or homogeneous time-resolved fluorescence assays selected as appropriate for a particular project target and with our clients. We express the ability of a PROTAC to induce target degradation as a DC50 value. On the right-hand side of this slide, we show data from a project targeting BRD4 degradation. Pharmacological inactivation of BRD4 is an anti-neoplastic approach that targets MYC oncogene expression. we have generated quality PROTACs that drive BRD4 degradation via a proteasome-dependent mechanism. We run multiple assays to assess a PROTAC's drug-like characteristics. For example, permeability across cell plasma membranes, as shown here, is critical for PROTAC activity. We focus on generating data from in vitro cell permeability, bioavailability, and transporter substrate assays to help inform the development of more drug-like PROTACs. Optimizing candidate bioavailability is key to candidate optimization for a successful project outcome. This slide shows a Medicilon case study for development of a PROTAC candidate and its backup, using the approaches that I've just gone through in the previous slides. First, key tool compounds were synthesized and project-specific assays established. In total, over 250 PROTACs were made for this project. Compounds were ranked on the basis of enzyme binding, cell inhibition, and protein degradation, and the best selected for DMPK and preliminary safety analysis. 10 compounds were selected for in vivo efficacy assessment, and the top compounds from this assessment were taken into a GLP in vivo evaluation of toxicity, enabling ranking of a lead and a selection of a backup candidate, both with appropriate characteristics for preclinical development. This project was delivered in only 10 months from start to completion, and this demonstrates our desire and drive to ensure that your projects are completed as rapidly and effectively as possible, to time, to budget, and to specification. Medicilon also has significant antibody-drug conjugate development capabilities and experience within the organisation. The three components of an ADC are the antibody that's responsible for binding to the target cell, the cytotoxic payload, a highly toxic compound, and the ligand, the linker, that couple the antibody to the toxin. Our biology team can generate novel antibodies de novo using traditional hybridoma approaches, phage display, or by sorting individual single B cells and sequencing them prior to establishing an expression line. Medicilon has produced candidates utilising its collection of over 50 toxins and over 240 ligands of linkers of varying types and design, but we can synthesize develop novel toxins, linkers, or antibodies as is needed for a specific project. Here are some of the toxins and linkers used by Medicilon. Medicilon can offer 5, 50 and 500 mg conjugation services. Experimental design cycles are in the order of 2 to 4 weeks for these antibody-drug conjugates, and a typical drug-to-antibody ratio of 2 to 8 is achieved. We undertake extensive quality control testing of your ADC or antibodies and generate significant certificates of analysis for you. Medicilon can characterize antibodies and/or antibody-drug conjugates in our preclinical development services to establish bioavailability, efficacy, and safety features for you. Medicilon also has a dedicated nucleic acid drug development team working on projects to develop antisense, siRNA, mRNA, and aptamer-based tools and therapeutics for clinical trials. We've developed an integrated platform approach that links effectively with our preclinical development activities. We've built a set of capabilities briefly listed here around the synthesis and modification of test compounds. For example, Medicilon has built an siRNA library with diverse monomers and a library of monomer synthesis blocks to rapidly complete synthesis of modified monomers. We've developed a range of analysis approaches, including the use of mass spectrometry, and we're well able to characterize your molecules completely for you. I've reviewed our candidate development activities and preclinical IND enabling services. But Medicilon also offers a range of services based around late-stage process development, formulation and manufacturing to deliver drug substance for clinical trials and API for marketed compounds. We have over 150 process development chemists and 100 formulation scientists that are dedicated to this work, and Medicilon's CMC testing support services can ensure the safety and regulatory compliance of our clients' products over the long term. Medicilon can provide analytical methods to generate your CMC, chemistry manufacturing control, data for regulatory submissions. We run a variety of chemistry analytical support assays and approaches for standardization of your product. We can look at inter-batch variation, we can set specifications, we can evaluate purity, contaminants, and look at compound chirality for you. Medicilon is available a broad range of techniques and approaches, including chromatography-based approaches, solid-state characterization analysis, and NMR. And if you're facing significant analytical challenges, I do recommend that you talk to our analytical teams. We have a dedicated chemistry team and dedicated laboratory workshops for process development, including both GMP and non-GMP environments. Our teams are experts in optimizing parameters to deliver GMP market-ready processes, optimized for cost, consistency, and ease of production. Our teams are expert in scaling and translating processes from early discovery into pharmaceutical GMP-grade manufacture at scale to supply clinical trials. We bring our experience early into the discovery process to optimize candidate selection as well as to deliver your high-quality API, and this de-risks your project, ensuring that you'll be able to continue with minimal challenges into the clinic. We bring our expertise and experience in process development and compound formulation early into the discovery process also. For formulation development, we are highly experienced across registration categories, from novel molecular entities to supplementary applications and generic development programs. We work across dosage forms, including solid, semi-solid, liquid, and novel dosing formulations, such as slow release and inhalation delivery systems. We work to develop formulations across the development stages, from candidate selection to new drug applications for the European Union, USA, and Chinese markets, as well as the UK. Our range of services include pre-formulation studies, formulation process development and scale-up, QC testing, and preparation of regulatory documents for applications to agencies. We believe that our manufacturing experience and capabilities can de-risk early phase projects, as well as providing manufacture of drug substance for clinical trials and products at scale. To deliver this, we have a GMP pilot plant that can produce batches of up to 50 kilograms of API drug substance, mostly for supply of clinical trials. Our main GMP manufacturing plant can make larger batches of up to 200 kilograms to supply very large trials or provide API and intermediates for the market. The expertise we develop in these facilities can help us to develop and progress your candidates more effectively in early discovery and preclinical phases. In my final slide sets, I discuss why I believe Medicilon can be an excellent partner of choice for your research and development projects and for your company. Medicilon offers a range of flexible service approaches to suit all customers, and Marshall will discuss these in more detail. We have a range of specific fee-for-service operations, offering a fixed price for a specific deliverable or set of deliverables. For more complex, for example integrated full drug discovery programs, we can price in terms of the number of FTE colleagues that are working on your project at any time. So here we charge for the number of FTEs working on your project, and this offers significant flexibility in terms of the size of the team that's on the project, the skill set it contains, and the prioritization of targets and projects within budget. We can offer a fully integrated service that includes milestone payments to minimize FTE costs and to deliver projects for you more cost effectively. Key to your project success is our selection of a top quality team with experienced project management and director level support for each project, facilitated by each and excellent communication. Benefits of partnering with Medicilon include high quality outputs delivered by a trusted partner. Over 100 preclinical development candidates have already been delivered. We have a track record of success. Medicilon has worked on over 260 candidates approved to enter clinical trials. We're fast, we have speed and flexibility on our side. We can have very short, up down to 7-day design synthesis and assay cycles for your development projects. We bring in early use of in vivo PK formulation and process development data to facilitate candidate selection and to understand your candidates better. And we have small molecule GMP API production analytical testing. Even if you don't need this early in your project, the experience that it brings can offer significant benefits for you. Most importantly, I think we have a critical mass of highly skilled scientific colleagues, well over 2,000 discovery scientists, and we have significant UK support via our Cambridge office to ensure that communication between yourselves and Medicilon is effective. We also bring, and this is important, problem-solving capabilities. Our quality teams, including experienced group leaders, project managers and directors, are experts in their field and can help you to solve problems when they occur and potentially even before they occur in your project. We have fully integrated capabilities from hit identification to clinical trials, and this means that we can spot those potential problems that might occur before they actually happen for you. As I've said, excellent communication is significant for us and for you. We bring written progress reports to your project. We have regular teleconferences that are designed to meet your specific customer needs. And we also have the potential for rapid communication via your preferred technology platform, whatever that might be. We have effective processes in place to resolve challenges rapidly, and we ensure that we deliver cost effectively for you. We have fully tailored, flexible business models based on years of experience. The IP that we generate for you is entirely owned by you and is well protected by Medicilon, and that's important. So this is my penultimate slide, and this really summarises the way that Medicilon can work for you from your initial idea through to IND and beyond. We provide fully integrated drug discovery where you can work in a virtual mode, allowing us to develop your drug. But we can also provide bespoke or established models, for example, an in vivo efficacy model to deliver a specific data set. And these assays and this type of mode of operating we can deliver very quickly for you. For early discovery projects, we are agnostic of therapeutic mode and have flexible service models to support customers by identifying and developing hits into leads. Medicilon identifies and optimizes lead series for small molecules and large molecules to make sure that your compounds have the right drug-like candidate properties. And we drive selection of candidates via strong data sets for efficacy, ADMET properties, non-GLP toxicity, and in vivo PK. Medicilon can facilitate access to the clinic via GLP toxicity safety, DMPK, formulation, process development, and stability testing. So in summary, my take-home message is that Medicilon is a full-service contract research organisation of high quality that can rapidly and cost-effectively support you to develop new therapeutics and achieve your commercial success.